This episode of the podcast is supported by Audible. You can download and listen to the world's best storytelling. I use it all the time to and from work. You can listen to audiobooks, original series, and more on their free app. To get your free 30-day subscription, which includes a free book, click on the link in our show notes and enjoy. Hey folks, welcome to the podcast. Today I had an amazing conversation with Dr. Güneş Taylor, and she's at the Crick Institute, and we talk about everything gene editing. So we talk about CRISPR-Cas9, which is a form of gene editing. We talk about all the cool stuff you can do with it, the risks, all of that stuff, and I hope you enjoy it. Hey, it's Lewis. Welcome to the podcast. Enjoy our conversations anytime, anywhere. Cool. I'm alive. Güneş, thanks so much for coming in.
Pleasure's mine.
We've just put the world to rights on politics.
Yeah, we really have.
But we'll move on to more interesting things. Gene editing. How did you get involved in that? How did it all come about?
The first I ever heard of gene editing was during my PhD. We were studying some cells that are pretty important during early embryonic development. And when you do experiments, you always need tools. Basically, you always need tools, molecular tools, and that was the first I ever heard of it when one other group leader came into a meeting of ours one day and said, "Have you heard of this stuff?" And we went away and read up on it, and I put my hand up and I said, "Well, I'd be willing to give it a crack and see whether or not it works." And so
nice.
that was the first.
And what was your PhD in?
I studied how the instructions that are involved in telling one particular gene what it's meant to be doing during really early embryonic development.
Wow.
And that gene's really important, and we know it's important because people who have a broken copy of that gene actually have a particular syndrome called charge syndrome. So we see that it results in what we call a phenotype. So people have a syndrome, basically.
Crazy.
Yeah.
And what you do this in, was it Nottingham University? Right?
So that was my undergrad, yeah. But I did my PhD at Oxford.
Cool.
Yeah.
How was that?
Long and hard, but very rewarding.
So the PhD was like 4 or 5 years?
Yeah, 4 and a bit.
Nice. And then, so now you're at the Crick Institute.
That's correct.
Cool. I'd never heard of it before.
Ooh.
But I Googled it when I knew I was going to speak to you, and it looks like it's doing some really cool stuff.
Yeah, it's I think the headline figure about it is it's the largest biomedical institution in Europe. It has like the most number of scientists under one single extremely shiny roof.
Wow. Where is it?
It's just around the back of the British Library by St Pancras. So 2 seconds away from the Eurostar.
Nice. So how does it work? So they just get loads of scientists under one big shiny roof, funded by
lots of different people. So the Wellcome Trust, there's the MRC, and then there's partnerships in with the universities, so Imperial, King's College, UCL. They all sort of both gave money, but also therefore have some of their group leaders in the Crick. And it's a really scientifically diverse space, and the idea is to really foster sort of innovation, collaboration.
And is there no kind of like agenda of kind of developing stuff to sell or like business outcomes or? So
no, it's a charity. We are supported on that side, so there is a really strong translation team. So anybody who wants to collaborate with companies to get like sort of molecules to test or develop pipelines, they're really encouraged to do that.
Yeah.
But it's not the explicit intention of the institution. The intention is just to do really great, relevant biomedical research.
Perfect for you.
Yeah, absolutely ideal.
And then did you go in there with a kind of agenda to research gene editing specifically? No,
so I went in to study about something called sex determination, which is basically to do with how do embryos develop into either males or females. And the gene editing part comes in because it's a tool, like I said to you. So to do really great experiments, you need really great tools, and gene editing, genome editing actually is the best way of doing a good, clean experiment. So
awesome.
Yeah.
So what is the technology?
Okay.
Exactly.
It's called genome editing as a bracket.
Yeah.
You can think of that a bit like mobile phones are a type of device, but there's lots of different types within that. And hopefully a fair few people might have heard about CRISPR-Cas9, which is the CRISPR word keeps getting thrown around at the moment.
I follow them on I follow some Instagram account that posts loads of stuff on CRISPR-9. Okay,
so exactly. CRISPR-Cas9 is a type of genome editing. So there are classically older types, a lot cruder, and the more recent one that occasionally has been used actually even in a clinical setting is something called a TALEN. And these are all just like complicated words, and if you go back to the phone analogy that I just gave you, CRISPR-Cas9 is kind of like the iPhone to mobile mobile phones, if that makes sense.
Yeah.
Right? So there's loads of different types, but
so like premium brands.
Exactly. We always knew when we got it, it's like the first time you ever saw an iPhone, you knew that was a game changer, basically.
Yeah.
So how does it work? As the name suggests, CRISPR-Cas9, it has two parts. One part is a single molecule that can be designed by the user, and it targets the Cas9 to whichever part of the genome that you're interested in. And the Cas9 is the business end, it's the doing end. The Cas9 cuts DNA.
Crazy.
And so you think, okay, so hang on, we've just said genome editing, so modifying genomes. How does one part user-defined sequence plus one part cutting DNA device work?
Yeah.
And how it works is DNA isn't meant to be cut. And when it does get cut or broken, and that happens sometimes naturally, cells have mechanisms to fix that. And those mechanisms sometimes have errors, make mistakes. And so we basically harness that by targeting the CRISPR-Cas9 to a certain place. We say, this gene here, I want it gone because I want to understand whether or not it's important to whatever process I'm studying. And so you send the CRISPR-Cas9 in, the Cas9 starts breaking that gene, and it gets repaired and it gets repaired. And then eventually the repair mechanisms slip up and a mistake gets made, which hopefully breaks the gene. And then it allows people like me to study what actually happens in the absence of a gene.
Oh, interesting.
And that's that principle of removing something or breaking it or messing with it in some way is how pretty much all of our science has come into existence, right?
Yeah. So how do you program? So you program a molecule to go into a specific part of the genome and cut it.
Yeah, that's exactly right. It's so funny these days. You order it online. You
order it online.
You just order it online. There's companies that sell you these single-stranded molecules.
Wow.
You use an online deposit to get the sequence, and then you just send an email.
Wow. And what, the Amazon next day delivery?
But borderline, yeah. IGT next day delivery. It turns up in an envelope, and you crack on with what you're doing.
Wow. And so what does it come? Is it like a liquid you just inject in someone?
No, not at all. So we get the tubes with the actual molecule in, and then you have to marry it.
And what state is the molecule in?
At that point, usually it's dry, so it's and you resuspend it, you add water or whatever it is you want, and then you have to bring the two parts together. So then you either have to...
So which parts do you...
So that's the CRISPR part that you order in, and then you need to bring in the Cas9 somehow to do the business end, right?
Fine.
So,
and then what exactly is the Cas9?
So the Cas9 is a protein.
Okay.
You have choices about how you introduce that into a cell. So you can either provide the DNA sequence to make that protein inside a cell, or you can get the protein itself and just give it to the molecule and then inject the whole complex the whole lot in. Yeah.
Wow, that's crazy.
Yeah, it is, isn't it?
Amazing. How long's it been going on for?
I want to say 2011, 2012. I think it was when I first started hearing about it. So yeah, good few years.
Wow. So I was reading. Have you read Homo Deus? It's a book.
Oh, Yuval Harari.
Yuval Harari, yeah. Human Gods and like the future of mankind. And it was interesting because part of that he was like, medicine's all about curing the sick, and then like the future or even the present is about improving the healthy. And you need, with these kind of things, it's like you can just very easily see so quickly how it could happen in a positive way.
Yeah. So it's important to say at this point. I mean, obviously, I'm completely partisan. I've always, as a scientist, you believe that what you're doing is meant to be used to improve life.
Yeah.
But it was really obvious from the very first time that we started reading about this that it's going to really challenge our society, basically, because now we've got to either put our money where our mouth is or not do that, depending on what people think.
Yeah. What's quite interesting, we digress a bit, so we'll go back to more of the technical stuff, but it's interesting to make sure that it's not just used for the wealthy, and it's it's equitable, and people in all parts of society get to benefit and stuff.
Exactly. It's like any technology. I mean, if you think even about IVF, right, which is a very related technology in some ways, it costs money, and then it becomes access restricted, basically.
Yeah.
And so that debate really has to start to
properly happen. Has it started yet, or not quite?
Yeah. No, definitely has started. Has started. The UN, all the way up to the UN and WHO are starting to at least acknowledge that this debate needs to happen and start saying we need to have more conversation and sort of trying to coordinate themselves, I think. Yeah,
interesting. And then so back to actually how it works. What can it be used for, or what is it currently being used for?
So a bit like when I said it was the first generation was like the first ever iPhone.
Yeah,
we are many moons down the line, and so now it can
like version 10 or
exactly iPhone 10x version of CRISPR-Cas9 now. So it can do a lot more than it used to be able to.
Right.
Like I said, the first version was just break DNA, and if you keep hitting the same spot, you'll get the break you want, the mutation you want eventually, and then you go from that, right? Now we can do things like provide repair templates, so we can deliberately break out parts of the genome and pop new bits in.
Wow. So cut and paste.
Exactly. There's other modifications where you can use the Cas9 to bring other molecules to a specific location, like a little carrier system.
And are these different techniques used for different applications?
Exactly. It depends entirely on what you want to do.
Sounds like you've got like a complete menu of whatever you want to do now. Amazing how quickly it's advanced.
Yeah, it really is incredible. I mean, and the best thing actually about it, and again, we shouldn't get too far into the sort of politics of it, but it's there's been a patent debate going on, or like argument really, going on in America, and the fact that it hasn't been patented has allowed it to be open source effectively up until this point. And
so the iPhone analogy is not great. Maybe we should use Android.
Yeah, maybe we should. Yeah, that's true. I just prefer iPhones. I think that's what it is. Whoops. Yeah.
So it's open source.
Yes. Yeah, you're absolutely right. It should be. It should.
So who invented it originally?
Well, okay. As with all sciences, it's kind of difficult to point the finger at the one person. There's two major groups, divisions. There's the Jennifer Doudna and Emmanuel Charpentier axis. That was one group at UC Berkeley, and then there's another group leader called Feng Zhang, who was at the Broad at MIT, and that's kind of the big division there, really. Okay. But they obviously built on many years worth of other people's research. So it's kind of like the glory of the relay, the last person who crosses the line. That's them. Yeah.
Oh, interesting. Okay, and then other research groups have taken it on and developed it. And
absolutely.
Fine. So you mentioned there's like a debate on patents. Is it really though? It's going to end up being open source in inverted commas and.
Well, so I mean, I hope so. Just as a user of this thing, but I don't know that it will be for sure. I mean, again, this is just my personal opinion, but I think it's a disgrace that anyone would even try and patent it, and this is what the debate's about basically, is this is a technology for modifying genomes. Everything that is alive on planet Earth has a genome, so if you patent that tool,
you're going to control
a lot of stuff. And I personally don't think it's particularly ethical to do so.
Yeah, no, absolutely. And so which way is the debate kind of going?
I honestly don't know. It's a very closed box.
Who's it between then? So it's
between MIT and Berkeley, as far as I'm
right.
aware.
Oh, okay, right.
Yeah. So it's between the two parties.
But then is one saying it should be open source and the others should be ours?
No, they're both saying
it should be ours.
Yeah, exactly.
And then everyone else is like, guys, just chill out. It should be everyone's.
In the meantime, everyone's like, they're distracted, keep working.
keep going, yeah.
Exactly.
And there's no kind of pharmaceutical company or anything involved as well, or?
I think there might be a spin-off company belonging to one of them or something like that. So I mean, obviously, so commercialization is a really interesting topic, basically, around this.
So if the debate is it's open source, then the really cool thing is every company can try and commercialize it, and hopefully the price comes down. And
then presumably, I mean, I'm no commercialization expert, but then what happens is you can have maybe even patent your singular application if you need to, or whatever, as a company. But it's a very different thing to have control over a very specific application versus the entire use of the technology.
Yeah, definitely. We touched on the kind of the safety aspect. So do you think we'd better kind of for humanity and society if it's open source and everyone's able to develop stuff rather than being controlled by?
So really interesting question, because the gut instinct is to say absolutely it should be, but then I also have to check myself and say to you, I don't know, have you heard about biohackers?
No, tell me.
Okay, so there is a group of people called biohackers. On the whole, they tend to be quite, the most vocal ones at least, are stateside,
right.
And these are people who really think that everybody, and they mean everybody, should have access to this technology and be able to just do whatever they want to do to themselves at any point in time. Exactly. So you raise your eyebrows and so exactly. So whilst I might say to you, yes, I don't think it should be patented and I think all humans should have access to this thing, I caveat that with, I do still think that it should be dispensed via established mechanisms that have been well researched and involve medical establishments.
No, definitely. It shouldn't just be like over or under the counter.
Yes, exactly.
I mean, you need to go into a hospital or wherever you might be able to get this kind of, Otherwise it's like, wow, our next door neighbour have just increased the IQ of their kid. I want to do the same thing.
Exactly.
Yeah.
And I mean, I also feel the urge now to say it's so easy to sort of oversell it like that. So you say, our neighbours just made their kid really intelligent. Well, the reason why it has to be done in a really controlled manner is because these traits, things like intelligence, are really, really complex and we don't know where it comes from. So you can't do that, and you can't tell people that they could do that and then give them the tool and send them on their way because they don't have the knowledge basically to really understand that they can't do that.
And once your DNA has been changed, all future generations you're gonna pass this on, right?
Yeah. So again, yes, theoretically, but I always have to remind people that the technology doesn't then magically disappear after. So technically, if I was to genetically modify my child, say, presumably CRISPR-Cas9 would still exist, and then if they wanted to modify the next generation, they could do that. But you're absolutely right. The point is, it becomes, under certain applications, it becomes inheritable. Yeah. Let's put it that way.
So, interesting. And then, so on the applications, so we touched on humans, which we can speak a little bit about, and other applications, agriculture, biotech.
Exactly. Yes, exactly. So as I said, genome editing technology can be used on any genome, technically, on planet Earth. So plants, animals and humans, each one of them technically has the same subdivision that humans do. So there's what's called somatic and germline. Germline is what we've just talked about. It's the one that is inherited because it is in your germ line, in your eggs and sperm, or whatever it is that you are as a plant or an animal. And then there's somatic, it's called, which is just your body, whether it's human, animal or plant. So there's different subdivisions of that. And absolutely, people are already, companies, not people on the whole, companies are starting to use this technology to investigate the use of genome editing technologies in crops, both in terms of yield, but actually these days increasingly with the sort of environmental lobby, there's actually mileage in trying to sort of broaden the range in which plants can survive. So with global warming coming, that could be really, really useful. Animals as well, if you can improve their yield without having to just pack them all into a smaller space, so you increase the nutrient density, say for example of an animal, then you get more bang for your buck and their welfare isn't compromised by overcrowding.
There's also quite a few companies that are growing meat in the lab to eat. That's really cool. Beyond Meat and all that stuff, right? It's really cool. Exactly. So that's a really cool interface between sort of tissue engineering actually and just pure research, and obviously genome editing tools can feed into that because then that's how you customize the cells that you're using to grow the meat. So I can say, right, I want a really beautiful steak and I want like loads of vitamin C, D, B12, whatever, and you could calcium, you can just whack it all in there.
Theoretically, theoretically.
Awesome. What application are you most excited about?
Oh, that's a trick question. No, so I mean, I'm torn now between saying obviously the applications of my research.
Of course. Which are?
As I said, I study sex determination, so I study how males and females come into existence. But what's really cool actually is I do that using chickens, and as you know, the chicken-based industries, eggs, for example, is one of the most heavily sex-biased in industry. So the male chicks always get killed, right, by the millions per year. And so one of the most exciting potential applications of my research would be to, for example, rig up a genetic system that means that the males don't get made in the first place. So I don't know if you heard recently there was a company from Berlin that was saying that like they'd bypass this issue of having to kill male chicks.
No, I didn't know. What's the story? The
story was that they've got a new platform to stop male chicks from being culled.
Right.
And actually when you look at it, what the technology does is it basically just sorts them before they hatch. So they sample some of the egg white before the eggs hatch, and then they can tell based on the hormonal content whether it's a male or a female, and then they dispose of those eggs. So it's a bit cleaner because you don't see the cute fluffy chicks, but the males still get made, right?
They still
So that would be one of the applications of my research, for example. So
awesome.
That would be nice and ethical.
But
as a human being, completely separate to all of this, and this is just my personal opinion, I think the most exciting applications are going to be when we're addressing human diseases that we know about. Single gene diseases, obviously obviously, things that we're really, really sure about. And there's only very few people on planet Earth that will be so affected that they have to use this technology. But I really think that you should put your money where your mouth is, and why shouldn't we use this? I think it's unethical to not use a tool that we have.
To cure disease, make us live longer.
Yeah, well, hopefully.
And then while we're living longer and our population grows, we can sort our agriculture out so we can get fed.
Yeah, I mean, the opportunities are endless. And then, I guess the last thing to say is obviously the most exciting part of this is the fact that if that's what we want to do with this technology as a society, and that's why this debate is so important, if we want to do that, then we have to get the research to actually lead us down that path to achieving that, because we can dream up any reality we want, but we then need to furnish that reality with knowledge.
Definitely. Right. And is the funding there, and do you feel it's going in that direction?
So that's a really interesting question. I mean, we're always underfunded and it's super competitive. I feel like perhaps companies are starting to realize that it's also a little bit unethical that all of the basic primary research is always funded by public institutions, and then once you have something that's promising, that's when a company
and the VCs come in.
Exactly.
So you've got the VCs. So how what's the ecosystem? You've got you guys and girls researching, doing some amazing stuff, and then the VCs are just circling around,
waiting,
meeting you for a coffee after the labs, waiting for stuff.
But things like that. I mean, like I said, different institutions have different mechanisms for encouraging us to come up with applications. But the way the funding works these days, a lot of the funding bodies obviously also want outputs, right? If it's publicly funded, you have to prove that you're doing something worthwhile. So whether it's a company or like a potential spin-off output, or whether it's promises of doing science communication, they're increasingly expecting us to do that, which is obviously entirely fair if you're
which
public.
Is fair. Yeah. I mean, also companies and businesses play a really important role, right? I mean, ultimately, if the money's there and it can be commercialized and it can start to have a really big impact, it's a great thing.
I mean, they're the experts in commercialization. So
they will get it out to the world, ultimately.
The masses, exactly.
Yeah, amazing chat. Thank you so much.
Thank you.
Pleasure. How can people find you and look at what you're up to?
I am on all of the social media platforms, obviously. So Instagram, Twitter. I have a website that you can email me through.
What's the website?
www.guneshtaylor.com.
Awesome.
Yeah.
Awesome.
So reach out, talk to me. I love this stuff, and I think it's really important that everyone starts talking about it.
No, thank you so much for coming in. It's really, really interesting, and we'll get you back in.
Yeah, please.
And
thank you.
Thanks so much.
Yeah, that's all right.
Hey folks, thanks for listening. Don't forget to subscribe in all the usual places.